And lastly, well, almost lastly, third one out of our four, proteins. Proteins make up a lot of our cell mass in the sense that they're structural. They're not just a chemical in solution, but it's what we're made up of. So our bones are made up of protein, collagen. I mentioned that. Our skin is made up of protein, collagen, keratin. Keratin is the protein that is a waterproofing protein. It gives us kind of a waxy layer, keeps the inside in and the outside out. So that's an important protein. Actin and myosin, if you remember that from general A&P, those are those contracting proteins that interact to form muscle contraction. Troponin is another enzyme that is a protein that is involved in muscle contraction. And when muscle cells, like heart muscle cells, are damaged, like as in a heart attack, the troponin enzyme is present in the blood. It shouldn't be in the blood. It should be in the muscle cell doing its job in the heart. But when the muscle cells of the heart are damaged, it releases troponin into the blood. And that's an indicator that doctors use, that they'll draw, the MLT, will draw a troponin and see if that is elevated, because it should be like 0.01. If a person has had a heart attack, then it might be as high as 4 or 5 or 6 or higher. It could even be less than that and still indicate a heart attack. So if you or someone you know is thinking, did she have a heart attack or did he have a heart attack? Say, what was the troponin level? Did they check a troponin? That's an early indicator of a heart attack. Okay, so remember that the polymer is a protein, so it's like a pearl necklace. We call the necklace the protein, and the individual monomers that make up that protein are amino acids, and they're bonded by peptide bonds. And the unique thing about proteins is they have a very specific shape, and that shape determines their function. If we change the shape, we can change the function, which is not good. So proteins are sensitive. They're sensitive to radiation, heat, excess cold, acids, bases. So if you look at a typical protein, there's two parts of it. There's an amine half and an acid half that has the COOH arrangement. We're not going to get a lot into the chemical structure, but you should just recognize it structurally, that it has this half and this half. All amino acids are the same, except for this R group is what varies from one amino acid to another. So there's 20 essential amino acids that we need in the human body, and they all differ just by this. So what's in the brown oval here is the same with every amino acid. It's this that differs from amino acid to amino acid. So again, taking two amino acids, putting them together, is a dehydration synthesis, forming a peptide bond there with the removal of water, and the opposite going in the other direction. Hydrolysis, where we're adding water and separating or breaking that peptide bond. So there's 4 levels of structure. I'm not going to hold you accountable for the details of this. I don't think this is relevant to how we care for our patients. Just know that the order of amino acids is one level of structure that's important. And then how those amino acids interact with one another is another level of structure that's important, and that's the hydrogen bonds between the different levels of the amino acids, or different types of amino acids. And then it folds in on itself, and there's other intramolecular forces that fold it in to give it a tertiary structure. And quaternary structure is when you have different parts of the whole compound that form a specific protein. Not all proteins depend on this level of folding in order to work, but an example of a protein that needs that level of folding to be just perfect and just right is hemoglobin. Hemoglobin is a protein that transports carbon dioxide and oxygen in the blood, and it has to have this very 4th level of structure, this quaternary structure, in order to function properly. People with sickle cell anemia have damaged quaternary structure because of a change in an amino acid. So if we go back to the primary structure, if I change an amino acid, that's going to change how that amino acid interacts to provide the secondary structure, which affects the tertiary structure, which affects the quaternary structure. So in low oxygen environments, people with sickle cell anemia, their red blood cells kind of warp, and they don't pass through the capillaries, and it causes clotting throughout the system, and they don't get blood flow to their extremities, and it's very painful. So they have to really be careful with their oxygen if they're traveling to high altitudes or exercising, or if they have bad lungs due to emphysema. they need a little more oxygen because they can get in a sickle cell crisis, which can lead to death eventually. So it's a serious disease, and it's an inherited disease that we'll talk about later in the semester, or in a couple weeks when we study genetics. So this is just a little review. You can tap on that if you want to, if you pull these up on your computer, about protein structure. Two major categories of proteins. So we had 4 different types of lipids. There's 2 different major categories of proteins. There's fibrous proteins, which are structural, and there's globular proteins, which have more of a function associated to them. So fibrous proteins are those that, like I say, give structure to the body. We talked about keratin in the skin, waterproofs the skin, gives you that waxy layer. Elastin gives flexibility to our connective tissues, allows them to bounce back when they're stretched. Collagen is the most abundant structural protein in the body. Like I said, we find it in a lot of our connective tissues in the skin. We find it in ligaments and tendons. We find it in bone. And then contractile fibers, we already mentioned those, actin, myosin. Those are examples of fibrous or structural proteins. Globular proteins are those that have very specific shapes. So some of them are going to exhibit that quaternary structure in order to function properly. And some of those include antibodies. So when you are exposed to some type of foreign body, what you call an antigen, you form antibodies. And the most common abundant antibody is IgG. So that's an example of a globular protein. So if you have a patient who has no appetite and is sick with an illness, low immune system, maybe they had surgery, so they had damage to structural proteins as well, right? So they need to heal. They need to build more structural proteins, collagen, right, to heal and span that incision. And then they have underlying disease, low immune function, and now it's time to order off the menu. What do we need to make these proteins? What does the body need to make new proteins? What has to come in from the diet? What? Protein. Yeah, protein. So when they say, I'll just have some strawberries, is that a good choice? And say, oh, you're healthy, you're ordering strawberries, good choice. If you don't know your advanced A&P, you might say that that's a good choice. And some oatmeal. I'll have some strawberries and some oatmeal. Is that enough to help heal and make more antibodies to fight infection? No, they need protein. And that's one of the biggest things, if you guys are nurses' aides today, that you can change tomorrow. Now that you know this, make sure your patients are ordering some protein on their menus. Say, well, let's talk about some protein. And one thing that my experience, people with a poor appetite and not a very good digestive tract, what they tend to like that are good protein sources are yogurt, cottage cheese with peaches, and like a hard-boiled egg. Those are things that the elderly eat that are palatable when they're not feeling well. When you say, how about a pork chop? No thanks, right? But even an omelet, oh, no thanks. Cheese, no thanks. When you're not feeling well, those things are not very palatable. But yogurt is a good one. So we need protein to make protein. Other proteins that are important, insulin is a very important protein. It helps lower our blood sugar. So we need that protein to keep our blood sugar stable and prevent blood sugar spikes. Cytokines and complement are 2 types of proteins that help our immune systems function. Cytokines activate the immune response. Complement promotes inflammation. EPO is another protein produced by the kidneys. When oxygen levels get low, it tells the bone marrow to make more red blood cells. Have you ever heard of blood doping before that some of the athletes were using? So they were removing some of their blood, stimulates the kidneys to produce EPO. The bone marrow makes more red blood cells, so now when they go to perform in their activity, they've got more red blood cells than the person next to them who did not blood dope. But now they're doing testing for that to make sure that people aren't doing that. They're actually measuring their hematocrit, their blood, the number of red blood cells they have in their blood, and they can't be different, during training and the time of when they perform. So we're trying to prevent that. Troponin I already talked about. Surfactant I already talked about. These are all globular proteins. And another one is renin. Renin is a protein-like substance that the kidney cells secrete when there's less stretch in the blood vessels leading to the kidney, and as a result, it raises blood pressure. So renin is a protein released by the kidney to raise blood pressure through a series of reactions, and we'll study that later on when we get into the urinary system. So because structure is so important to protein function, denaturing is an example of, or is a process in which we break protein structure. So to denature a protein means you're changing the structure, and that can change the function. So there's a number of things that can cause denaturation. Simplest example you can think of, next time you cook an egg, that egg yolk is a protein. When you add heat to it, it becomes solid white. You can't go back and get that slimy, see-through egg yolk, or not egg yolk, egg white again. It's been denatured. That protein is completely different structure, different appearance. And that can happen in our bodies as well. We can have permanent reversals of tissues that we can't bring back because the proteins in that tissue have been denatured by a burn, for example, a chemical burn. So enzymes, the key thing to know here is that all enzymes are proteins. So if it's an enzyme, it's a protein. So all enzymes are proteins. And we can proceed with all of our metabolic reactions in the body at 98.6 because of enzymes. Enzymes lower the energy for reactions to occur, and because of enzymes, we can maintain at 98.6 and not have to have a higher temperature to maintain that same metabolism. Now what happens, again, to proteins if we raise the temperature? They denature. So if we have these enzymes that are, producing neurotransmitters and keeping our homeostasis in check, and we get a fever of 106, that is going to denature proteins, and that's going to affect metabolism and nervous system function. And a symptom of a really high fever in kids especially is being lethargic, being really, really sleepy. So it's always important to watch kids that are getting a temperature as bedtime approaches, because we know that temperatures rise through the night, and there have been unfortunate incidents where parents have had a baby, say, toddler, a 2-year-old or 1-year-old with a fever of 100, 203, and they say, well, let's just see how it works out in the morning and just put baby to bed, and baby passes away overnight of a febrile seizure. So if you have a child that's going to bed with a fever over 101, and you're not treating it and seeing that fever come down, then you should keep that child near you so you can check on the child throughout the night and make sure that fever is not climbing, because that's going to change the proteins in the body and could lead to death, seizures and death. I know someone in our community that happened to, actually. Child was sick, tucked the little boy in bed, and the next morning he had passed away through the night. Perfectly healthy child, just had a virus and had a febrile seizure through the night and passed away. Very, very sad. Okay, so enzymes, I want to just refer you to this diagram. I'm not going to go into a lot of detail on enzyme activity, but you should know that the substrate is whatever chemical we're acting on. So if I bring in a cracker that's full of starch, starch is the substrate, and an enzyme is going to act on it. And the enzyme in our saliva that acts on starches is called amylase. So enzyme E here would be amylase, that when that starch, in this case it's amino acids, but starch is what? What is starch, did we say? Carbohydrate. What type of carbohydrate? Mono, di, or polysaccharide? Do you remember? Starch. Is it a simple carbohydrate, or is it a long storage? Yeah, so it's a poly. Poly means many. So it's a polysaccharide. So when it bonds to the enzyme is going to break those covalent bonds between those carbons in that starch and release, well, this one's going the other way. This is too bad. Okay, well, I can still use my example. And then it would break those bonds and release those little sugar units, and that can be digested then. The example here in this slide is taking 2 amino acids, interacting with the enzyme, and that enzyme is forming a peptide bond, releasing water. Again, so this is what? Dehydration, synthesis, dehydrating, losing water, and forming something bigger, which is a dipeptide, or 2 amino acids bonded together. So the enzyme, does the enzyme change in the process? No. So enzymes are found in small amounts in the blood. They don't change in amount because they can be used over and over again. But they're very specific. Notice that active site has to exactly match the molecular structure of the substrate of those amino acids in order for that reaction to occur. So enzymes are very specific. And then lastly, DNA and RNA. We're going to talk about this a lot in labs, so I'm not going to belabor this too much. But the key thing you need to know is the building block of DNA and RNA are nucleotides. And a nucleotide is made up of a phosphate, a sugar, and a nitrogen base. So there's 3 parts to a nucleotide, and we see repeating units of these 3 pieces that makes up DNA. So 1/2 of the DNA molecule contains nucleotides with a specific nitrogen base arrangement, and the other half has a complementary arrangement of nitrogen bases. And again, we're going to talk more about this in labs, so I don't want to belabor this. We've been sitting a long time here. But the key thing to know is that the DNA molecule is a double-stranded, twisted ladder. And the purple and the light purple on the side are phosphate and sugars. And the sugar in DNA is deoxyribose. And what forms the rungs of the DNA ladder are these nitrogen bases. RNA is similar, except the sugar in RNA is ribose. Thymine is a nitrogen base in DNA. We replace that with uracil. And it's a single-stranded molecule. So here's DNA, and RNA is single-stranded. So we only see basically like 1/2 of what we would see in a DNA ladder. So here's the nitrogen bases going down the center, and we'll talk more about those in lab. And lastly, ATP. This is the fuel for our cells. So you go to the store, you go to the gas station, you give them money to give you gas, and gas is what helps your car go, right? So when we think of cells, what helps our cells transport substances across the membrane, contract our muscles, that movement of structural proteins and movement of substances across the membrane is because of ATP fueling that process. I can't stuff dollar bills into my gas tank and make my car go, right? So the dollar bills is like glucose. We need glucose to come in to make ATP through the process of metabolism, and that's next week's topic of how we go from glucose to ATP. So for now, what you just need to know is that glucose is the energy, is the fuel that starts the process to get ATP, but ATP is the energy currency of our cells. It's what our cells directly use to get energy. And what's special about ATP are these little red bonds between the phosphates. So there's energy. Think of these like a coiled spring that when I break these bonds, I release the energy there, and that fuels a process. So we take a phosphate from ATP and we add that phosphate with that special bond to another molecule. We call that phosphorylation. If I remove a phosphate, like if I remove a phosphate from ATP, we call it dephosphorylation. So we can move energy back and forth. When we form ATP, we're storing energy. When we break ATP down, we're releasing energy. So there's ATP, which means how many phosphates? 3. Then we have ADP, 2 phosphates, AMP. Which has the least amount of energy of these 3? AMP, right? Less phosphate groups. And which one has the most energy? ATP, okay? So basically ATP, the structure, is the amino acid adenine bonded to a ribose sugar that we see in RNA with 3 phosphate groups. So that's ATP. So this is a reversible reaction. Again, when we're going to the right, when we're breaking down ATP, we're releasing energy. When we're forming ATP going in this direction, we're storing energy. And again, it's a dehydration, synthesis, and hydrolysis reaction. So in summary, looking at these major organic molecules, whether it's proteins, lipids, nucleic acids, or carbohydrates, there are certain foods that are rich in these molecules. So if a person wants to have a healthy blood sugar, let's say they have insulin or they're getting close to being diabetic, they have pre-diabetes, they don't want to have excesses of carbohydrates, because if you look on your right-hand side, my left, you can see obesity, diabetes, cavities, intestinal irritation, high triglycerides if we have too many carbohydrates in our diet. So we really have to watch our balance of these nutrients. And then if you don't have enough, then obviously you're not going to have enough energy to fuel your processes, so the body burns fat. And when the body burns only fat for energy, it causes acids to be produced. And we'll study that with metabolism next week. Have you heard of ketoacidosis? Or people that go on the Atkins diet, that they eat eggs and bacon, and my brother and my husband both did this years ago, and they lost like 20 pounds. But all they ate was fatty foods and no carbohydrates. Because if you don't have any carbohydrates, your body is primed to go after fat to get the energy it needs and make glucose from that. And we'll study that next week. But not good for the body long term. It's very hard on the kidneys. Again, lipids, we can see there's different types. Omega-6s, omega-3s. Like I said, we have to be careful with some of those omega-6s because they promote inflammation. Omega-3s are the better ones. And then the proteins. So there's complete proteins and incomplete proteins. Who has to worry about proteins, do you think? What type of person out there with a what? Vegetarians, yeah. They really have to be careful that they get enough protein in their diet, because animal proteins are complete proteins, but vegetables and seeds are incomplete proteins. So they have to combine certain proteins to make a complete protein to get enough of those amino acids that are necessary for life. Like I said, there's 20 essential amino acids we need for life. We need to get those in our diet. The pieces anyway, and then we can assemble them. Okay.